Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome to the next lecture in the third unit of Plant Biotechnology course. In this unit, we are learning about the plant protoplasts, how these can be used or are being used to study the basic biological phenomena in plant sciences and several commercial applications for the crop improvement or generating new cultivars. My name is Manoj Sharma and I am working as an assistant professor of plant biology at Jawaharlal Nehru University. In JNU, I teach plant biotechnology and genetic engineering at School of Biotechnology. These lectures have been reviewed by Professor Kashmir Singh, who is working as a professor of plant biology at Department of Biotechnology, Punjab University, Chandigarh. This project is sponsored by DTH Swayamprabha, MHRD, New Delhi. So in this lecture, I will talk about the various methods used for selecting the hybrid protoplasts and how to culture these protoplasts for regeneration. Then I will discuss about the various methods that can be used for the characterization of the hybrid plants, and finally the advantages as well as the limitations of the somatic hybridization. So first, a quick recap of the last lecture where we started to talk about the somatic hybridization. We learned about the somatic hybridization in journal. Then we talked about various methods of protoplast fusion, where I discussed about the spontaneous fusion of the protoplast or the use of the mechanical force to create the fusions. Then we discussed about the role of various fusion-inducing chemicals and the use of the electric field for the induction of the protoplast fusion. We specifically learned about how the polyethylene glycol or the calcium ions in the alkaline condition can be used for induction of the protoplast fusion. I also talked about the various segregation patterns of the cytoplasmic genome, that is the chloroplast genome or the mitochondrial genome, when the hybrid cell divides post-hybridization and is cultured on the in vitro media for regeneration. Finally, towards the end of the lecture, I talked about the different types of hybrids like the symmetric or asymmetric hybrids, and now we will continue our discussion further with the remaining aspects of the somatic hybridization. So once the fusion of the protoplast has been performed, next step is the selection of hybrids or heterokaryocytes from the hybridization mixture. If we are performing the microelectrofusion, we talked about it in detail in last lecture, where one-to-one fusion of the protoplast takes place. The selection is not a problem because only a single fusion body is formed and it can be directly transferred onto a suitable culture medium for regeneration. However, usually the fusion suspension is a heterogeneous mixture of heterokaryocytes, homokaryocytes, unfused parent protoplasts, or even the organelles like the chloroplast may be there. In fact, the frequency of the desirable products, that is the heterokaryocytes or heterokaryons, will be much lower as compared to the other products. Therefore, identification and separation of the hybrids is one of the major challenge for success of the somatic hybridization. Several strategies have been used in the past for this purpose, but none of them is universal, which can be applied to all the plant species or any kind of protoplast fusion. Here is a list of various type of markers or methods that have been used for selecting the hybrids. First one is the visual markers like the pigmentation or the color. Then we have the biochemical methods and genetic complementation. So the first one is the visual marker. Now the hybrid cells may have some visual characteristics that are distinct from either of the parent. For example, fusion of the pigmented and non-pigmented protoplast, or the hybridization of the protoplast having different colored pigments. Here are the two protoplasts that have clearly distinct pigments that gives a characteristic color to the protoplasts, or these have the contrasting visible phenotypes. During the hybridization of these protoplasts having the contrasting color phenotypes, their cytoplasm will fuse together and the mixing of the cytoplasm will occur, and hence the hybrids may not be able to retain any particular color, rather these may have an intermediate color, and this can be identified in the culture by analyzing the hybrids under the microscope. Similarly, the protoplast can be given a visual phenotype artificially by staining the protoplast maybe with the different kind of dyes like fluorescent dyes or other type of staining techniques can be used to label the protoplast before the fusion. And mixing of the dyes in the hybrid can be used for tracing the hybridity or hybrids under the microscope. So the next is the biochemical complementation. Depending upon the characteristics of the protoplast used in fusion, various biochemical compounds can be used for selection of desired hybrids. Presence or absence of these compounds select the hybrids based upon the differences in the expression of a character by the hybrid cells, which will not be there in any of the parent protoplasts. Like the auxin sensitivity. It has been observed that in some plant species like the legumes, the protoplasts are unable to proliferate on the auxin-free medium. Here are two protoplasts from the legume species where auxins are required for the proliferation. That means in order to multiply these protoplasts, exogenous supply of the auxins would be required, and if these are cultured on the auxin-free media, they will simply not grow. However, when the fusion occurs between these protoplasts, hybrids are able to grow without the exogenous supply of the auxins. That means from the hybridization mixture of these two protoplasts, only the heterokaryons would be able to grow on the culture medium without the auxins. Similarly, chlorophyll complementation for the hybrid selection in the light-sensitive tobacco albino mutants has been used. In few tobacco cultivars, albino mutants which lack the chlorophyll are sensitive to light. That means they need to be incubated in the dark for their proliferation, and in the presence of light they will simply not grow. However, the hybridization of these protoplasts abolishes their light sensitivity and chlorophyll development also starts. Hence, the complementation between the parents mutually abolish the deficiency, and the presence of light can be used to allow the proliferation of only hybrids in this case. However, these are plant-specific characters and are not applicable universally. But if such kind of traits are available for any plant species in question where we are going to perform the hybridization, these are very useful. Similarly, other traits like resistance to antibiotics or herbicides, use of amino acid analogs or phytotoxins can be used for hybrid selection in the similar manner. Next is the use of metabolic inhibitors. Several chemicals are available that can interfere with the functions of the cell. Some of these chemicals can inactivate the cytoplasm irreversibly and inhibit it from the further division, like the use of iodoacetamide or rhodamine 6G. Though the mechanism of their action are not clearly understood, however, iodoacetamide is an irreversible inhibitor of the cysteine peptidases, and rhodamine G is a potent inhibitor of oxidative phosphorylation, and their application inhibit the cell division. They do not let the cell to divide and grow further. So here is a protoplast. When it is treated with the metabolic inhibitor iodoacetamide, in this case, these protoplasts are not able to divide further. Iodoacetamide inhibit the cell division, though the mechanistic details are not very clear. Its effect is dose dependent. That means the concentration required to inhibit the cell division may vary among different plant species. Like for the cotton, lower concentration of iodoacetamide, concentration of about 0.5 millimolar, can inhibit the cell division. However, in order to stop the proliferation of the citrus cells, at least six-fold higher concentration of iodoacetamide would be required. The fusion of the iodoacetamide treated protoplasts with the non-treated protoplast can restore the cell division capabilities. Similarly, the protoplast of the other partner may have some other characteristic features, like it may have an auxotropic mutant, so it would not be able to grow on the media without the exogenous supply of a particular supplement. However, when the protoplast, that is iodoacetamide treated protoplast, and the protoplast from the auxotropic mutants are fused together, these deficiencies may be complemented by each other, and the hybrid cells may be able to grow and divide on the culture medium without the supplement of which are required by the auxotropic mutants. And in this way, only the hybrid will proliferate on this media. Finally, the hybrids can be transferred onto the suitable culture media and hybrid plants can be regenerated. Next is the genetic complementation. Now, the genetic complementation is another method for the selection of hybrids or regulate the genomic contribution to the hybrid to some extent. Basically, the genetic material of one of the parent may be fragmented and removed. Suppose we have two plant species, that is species A and species B, and we want to combine the cytoplasmic genome of the species A with the nuclear genome of species B in the hybrids. So the first important point is that we don't want the nuclear contribution from the species A. Therefore, we can use the genome fragmentation tools to destroy or fragment the nuclear genome of the parent A, and it is usually done through the radiations, and we will learn about it shortly later in this lecture. Now, these protoplasts without the nucleus would not be able to replicate, and when these will hybridize with the another protoplast, these will not contribute anything with respect to the nuclear genome. Now, from the species B, we want the nuclear genome, and hence now we can combine the metabolic inhibition of the cytoplasm in this case. So the protoplast of the species B can be treated with the metabolic inhibitors like the iodoacetamide. The iodoacetamide treated protoplast will not be able to replicate again, and as it causes the irreversible cytoplasmic inhibitions. However, when these protoplasts will fuse with any other protoplasts, these would be able to contribute the nuclear genome. Therefore, when the UV irradiated protoplasts of parent A, where the nuclear genome have been fragmented, will fuse with the iodoacetamide treated protoplast of parent B, where the cytoplasm has been inhibited, it can restore the cell division and growth capabilities in the hybrid only, and only the desired hybrid would be able to regenerate or grow in the in vitro cultures, and this makes the selection easier. Finally, the hybrids can be transferred onto the suitable culture media and hybrid plants can be regenerated. In order to obtain the asymmetric hybrids, genome of the one of the parents needs to be removed partially, or it needs to be defragmented. It may take place automatically after the hybridization because of the genomic imbalance or because of other reasons when we hybridize the protoplast from very distant varieties or distant species. However, before hybridization also, genome can be fragmented or partially eliminated by application or by use of some biotechnological tools. Various techniques have been used for this purpose. However, the major ones are the irradiation or the induction of the microprotoplast preparation. So the first is the irradiation. Now, the genome fragmentation can be achieved by the exposure of the protoplast to radiations, which usually cause the DNA or the chromosomal breaks. Commonly used radiations for this purpose are either ionizing radiations like X-rays or the gamma rays, or the non-ionizing radiations like the UV rays. Exposure of the cells or the protoplasts to the radiations causes random chromosomal breaks, and it can be used effectively for generating the asymmetric hybrids. In addition to the DNA damage, radiations may cause other negative effects on the protoplast that may reduce the protoplast's viability. Therefore, the dose of the radiation used for the genome fragmentation or to damage the nuclear genome is very, very critical. Further, the radiation susceptibility of the protoplasts prepared from different plant species also vary greatly, and hence optimization would be required to identify the optimal radiation dose for genome fragmentation without compromising the protoplast viability. Though both type of radiation have been used successfully to generate the symmetric hybrids, however, the non-ionizing radiations like the UV radiations are preferred over the ionizing radiations because of their application is relatively easier. The ionic radiations further may cause the unintended effects and compromise the protoplast viability. Further, the reproducibility among the various experiments is also much better in case of the non-ionizing radiations as compared to the use of ionizing radiations. Next, or the second method of genome fragmentation is the microprotoplast preparation. Earlier, while we were talking about the plasmolysis and the protoplast preparation, we learned about the formation of different types of subprotoplasts that may be formed while isolating the protoplast. There are several categories of the subprotoplasts, and one of them was microprotoplasts. The subprotoplasts that carry the fraction of the nuclear genome along with the cytoplasmic content are called as microprotoplasts. It means that the microprotoplasts carry partial genomes or the partial nuclear genome. However, the content as well as the type of the DNA packed in the different subprotoplasts varies from one microprotoplast to another. The production of such subprotoplasts can be induced artificially. Like the micronuclei production can be induced artificially using the spindle toxins. The micronuclei are the small extranuclear bodies that arise during the nuclear division, and like the nucleus, these contain the DNA. However, they usually contain the smaller chromosomal segments or the fragments or the chromatids. So basically, they contain or they carry the part of the genomic DNA. The distribution of the nuclear material in the multiple smaller structure can be induced by the use of these spindle toxins. Usually, the toxic substances like the antimitotic herbicides or the colchicines, that is the spindle toxins, are used for this purpose. These are applied at the metaphase in a synchronized culture. Application of these toxins inhibit the metaphase, which result in the scattering of all the chromosomes in the cytoplasm. So here is a cell which is ready to divide, and it undergo metaphase and cell division. However, if the spindle toxins are applied in the synchronized culture, that is, all the cells in the culture are in the same stage of division, spindle formation will not take place and all the chromosome gets scattered in the cytoplasm. Later, these scattered genomic fragments or the chromosomes decondense into a smaller nuclear-like bodies and are called as the micronuclei. The efficiency of these chemicals largely depend upon the type of chemical, the size of the dose, the time period for which the protoplasts are exposed to these chemicals, and finally the plant genotype too. The response of or the sensitivity of the different plant species to these chemicals also varies. Therefore, parameters may need to be optimized in a species specific manner. Now, these cells are processed for the removal of the cell walls using the enzymatic methods and induced to make the microprotoplasts. Usually, ultracentrifugation can be used for this purpose. Further, these microprotoplasts may be filtered through the multiple sequential filters of the different pore size to separate the microprotoplasts of the different sizes. The next is the hybrid culture and regeneration. So once the selection have been made to select the heterokaryons, the next step is to culture these heterokaryons and regenerate the hybrid plants in vitro culture conditions. So the first step is the formation of cell wall around these hybrid protoplasts. Now, before the actual cell division, protoplasts usually increase in size and undergo general division within first few days, usually within the first week. Once the cell wall is formed, it becomes a complete cell, and after the formation of the cell wall, these hybrid cells lose the spherical shape too. By the third week of their culture, small visible colonies are formed, which can be used for the initiation of the callus development. Once the callus induction is initiated, the usual regeneration protocol is followed for the organogenesis or the somatic embryogenesis. As we discussed earlier in the unit 1, the various parameters of in vitro cultures. So, and in this way, hybrid plants can be regenerated. So therefore, in order to perform the somatic hybridization and regeneration of the hybrid plants successfully from these hybridization experiments, we need to have an optimized regeneration protocol for these plant species. And if the in vitro regeneration protocol is not available, and if you want to perform the somatic hybridization, we should first focus on optimizing an efficient regeneration protocol for these plant species. So the next step is the characterization of the hybrid plants after the regeneration. So now we have selected the hybrids, we cultured them on the in vitro conditions, regenerated the shoots, and developed the plantlets. So we have the hybrid plants now. However, the characterization of these plants and or the confirmation of their hybridity will be required. So hybrid plants may be identified based upon several different types of features. The first one is the phenotypic characters. Now, the hybrid plants are usually intermediate between their parents, and therefore often they have intermediate morphological characters too. Usually the vegetative characters like leaf shape or area, morphology of the roots, etc., or the flower characters like size of the flower or the color of the flower or the structure and shape of the flowers, These are some of the most commonly used morphological characters to study in the hybrid plants, and they may have intermediate phenotypes between their parents' phenotype. Although the genetic basis of these morphologic variations or differences are largely unknown. Then next is the chromosomal constitution. So basically the counting of the chromosome can be performed. The total number of the chromosomes can be counted directly using the chromosomal markers, and it can provide the information about the ploidy level that can indicate if it is just a normal parent clone or if it is a asymmetric or a symmetric hybrid. And finally is the use of molecular markers. Usually these are based upon either the protein or the DNA. Like the most commonly protein-based molecular analysis used to identify the hybrid plants is the isozyme analysis. So the isozymes are the multiple molecular forms of the same enzyme and can execute the same functions too. These enzymes may have cultivar-specific electrophoretic bending patterns and can be used to check the hybridity. So basically, electrophoretic separation profiles of the isozyme are generated for the parents as well as the hybrids, and then these are compared and hybrids can be identified in this way. Some of the enzymes that have been used for this purpose in the past include amylases, peroxidases, esterases, or even the alcohol dehydrogenases. And the other type is DNA-based markers. The use of DNA-based markers can generate important genetic information. Several different kinds of markers are available. Earlier, RFLPs and RAPDs were more popular. PCR-based markers are still popular. They are easy to handle and can be easily used to distinguish the hybrids from their parents. However, because of the sharp drop in the sequencing cost over the last few years, even the sequencing-based analysis can be performed for the characterization of the hybrid plants. So in this way, these are some of the important tools that can be used for the characterization or the identification of the hybrid plants. So now the next is the advantages of the somatic hybridization. The first one is that it can overcome the sexual incompatibility and production of novel interspecific or intergeneric hybrids can be achieved. Distantly related species are reproductively isolated and do not allow the genetic exchange among the species. Therefore, due to the reproductive incompatibilities, the agronomically important genes cannot be transferred across the sexually incompatible plant species. Like if you want to transfer a trait from a species from one genera to another genera, it would not be possible through the sexual hybridization or through the sexual reproduction. However, the somatic hybridization provides an opportunity to overcome these sexual incompatibilities. Several interspecific, intergeneric, or even intertribal or even the interfamily somatic hybrids have been produced successfully. The next is the novel combinations of cytoplasmic and nuclear genome can be formed. So in the sexual reproduction, cytoplasmic organelle are generally contributed by only the female parent, and therefore all the cytoplasmic genes follow the maternal inheritance. However, somatic hybridization again provides an opportunity for transfer of the cytoplasmic genomes like the plastid genome or the mitochondrial genome from any of the parent to the progeny plants. These novel cytoplasmic combinations can be made only through the cytoplasmic hybridization, or it is also called the hybridization. In addition, it can also be used for creating novel combinations of the cytoplasmic genomes, like the chloroplast genome of one parent or the mitochondrial genome of another parent can be combined together in the progeny. So basically, it also provides an opportunity to mix the cytoplasmic genome from the different plant species. Again, this is the only method where such kind of combinations can be generated. In addition, it can also generate the recombinant organelle genomes. Like during the somatic hybridization, when the protoplast from the two different parent fuse to form the heterokaryons, these fusing bodies usually have the organelle from both the parents. And then slowly the organelles are selectively lost or eliminated with the further divisions or their regeneration. Now, during this time, when these organelles are together in the fusion bodies and or at the earlier stages of the division, exchange of the genetic material between the cytoplasmic genome can occur. And specifically, it is very frequent in the mitochondrial genome. And therefore, the hybrid plants may have the mitochondrial genome that do not belong purely to any one of the parent. Plastid genome usually do not interact or exchange the genetic material even when they are present or even when the plastids from two different plant species, they are present in the same cell, that is the heterokaryon, for some time. And usually these are purely belong to one of the parent involved in the fusion. Somatic hybridization is a good system to study the fate of plasma genes and help in transferring the cytoplasmic characters, that is the traits that are controlled by the cytoplasmic genes located in the either the mitochondrial genome or the plastid genomes, like the cytoplasmic male sterility or several traits associated with the photosynthesis. They may be transferred from the one species to another species, or novel combination can be generated for these kind of characters too. Further, the mixing of the nuclear genome opens up the door of the transfer of traits and to study their expression, their interaction, and the stability, and the genetic changes that are associated with these new combinations of the genes can be generated in this way. Somatic hybrids can be used for the developments of the fertile diploids or the polyploids from the sterile haploids or triploids. Further, novel somatic hybridizations can be performed to generate the seedless fruits, as has been done for the citrus, where the haploid and diploid protoplasts were fused to regenerate the triploid hybrids that had seedless fruits. Further, it can shorten the breeding cycle, as the somatic hybridization can be performed on the plants that are in their juvenile phase. Somatic hybridization has been successfully used to generate or transfer of the genes encoding for the important traits like disease resistance, tolerance to the abiotic stress conditions or the environmental conditions, or the tolerance to the herbicides. So in this way, several improved hybrids for these traits have been generated in different plants like citrus, solanum species, brassica, tobacco, and so on. So we talked about a lot of positive factors associated with the somatic hybridization. or various type of the opportunities that somatic hybridization provide. However, there are several challenges that need to be taken care. So theoretically, somatic hybridization is very attractive. However, there are several challenges, like the plants regenerated from the somatic hybrids are not always fertile, and they may not make seeds at all. Though the fusion of the protoplasts from different species or different genera, that is the plant species which are distantly related phylogenetically, it is easily or can be performed easily, but the production of the viable hybrids or the regeneration of the hybrid plants may not be possible in most of the instances. When the protoplasts are produced from the taxonomically different plant species, the somatic hybrids are usually unbalanced as far as the genomic content is concerned, and they may not be viable, or even if they are viable, they may have very poor regeneration capabilities. Many of the somatic hybrids that are successfully produced and are fertile may not be useful, as there is no way which can regulate the expression of any particular specific character in the hybrid plants. The identification and the isolation of the hybrids from the pool of the hybridization mixture at the cultural level is a major challenge. Though we discussed several methods for the selection of hybrids earlier in this presentation, however, the standardized protocols for selecting or identifying the hybrids are not available that could be applied universally to all the species. So in summary, we learned about the various methods of hybrid selection that are used for selecting the heterokaryocytes. However, we also learned that these methods cannot be applied universally for the selection of somatic hybrids from any of the plant species. Hence, this step of the hybrid selection is one of the major challenges for successful somatic hybridization. Then we talked about the culturing of the hybrids in vitro conditions and regenerating the hybrid plants. We also discussed about the various tools that can be used for characterization or identification of the hybrid plants. And finally, we discussed about the advantages or the opportunities that the somatic hybridization can provide, as well as the areas of limitation that need to be worked out. So overall, somatic hybridization is an important tool for the crop improvement, and it can be used to produce hybrids with the novel genomic combinations that cannot be generated through the other sexual hybridization methods. So it has a huge potential. However, there are several areas that need to be worked out in order to use it efficiently for the crop improvement across all the plant groups. Thank you.